Now, by a show of hands, I need to know, guys, like be truthful. Raise up your hand if you are struggling and you just don't understand what I'm saying. Okay. What is
Wait, Miss,
confusing you?
have I like been unmuted this whole time?
Yes,
Oh, I'm
but I'll
sorry.
mute you. It's okay. So, what part is confusing you, kiddo? That way I can see how I can help you.
The strands of DNA is what's confusing me.
Yeah, that's really confusing, right? Because I'm talking about these strands, but you have no idea what I'm talking about, right? And that's good. I'm glad that you brought that up because I'm about to show you an example and we're going to do it together. And hopefully this will kind of clear up what I mean by strands of DNA. Okay? So I'll ask you again at the end to see if that kind of helped. If it didn't, then I'll figure out a different way to teach it. So the next part we're going to do together is this part that says practice. So this is what you guys are going to fill out. Now, I'm going to be showing it on here. If you look, they're exactly the same. I'm going to do it on here, and you guys are going to do it on here. So whatever I do, you're going to do. First off, guys, I want to follow my 3 steps. The first step is I want to figure out what restriction enzyme I'm using. So the restriction enzyme we're using today is called HALE. And I'm going to spell it over here. So the restriction enzyme we are using today is called HALE. And HALE cuts DNA in this specific base pairs. So what this means is like, imagine, that's like me giving you instructions and saying, hey, I need you to cut this piece of paper, but you need to cut it right here in the middle. I don't want to cut anywhere else, but right here in the middle. And then you look and you're like, okay, I'm going to cut this paper right here in the middle. And then I say, this paper right here, I want you to cut it right here in the middle. And you keep on cutting the pages right in the middle like I instructed. That is how restriction enzyme works. It's very specific and it tells you exactly where it wants you to cut. So this restriction enzyme, HALE, wants you to cut between GGCC. And I'm going to show you guys what that means. So this restriction enzyme wants you to cut between GGCC. So right here, I have my main sample. So we're going to pretend this is DNA found at a crime scene. Now, the first thing is if I look at it, I have 1 strand of DNA. Now, I want to know, do you guys think DNA has 1 strand of DNA or 2? What do you guys think? So once again, guys, does DNA have 1 strand or does DNA have 2 strands of DNA?
Wait, if DNA has what?
Okay, so when you think about DNA, does DNA have 1 strand or is DNA made up of 2 strands of genetic information?
2.
It's made up of 2. Very good. That's why it's called a double helix, because 1 strand goes this way, the other strand goes this way. So it's called a double helix because you have 2 strands of DNA. But if I look at this here, how many strands is that?
1.
This is only 1. So I need to figure out what the second strand is. So this is how I figure out. What pairs up with G? So I'm looking at this first letter here. What pairs up with G?
C.
Very good. So this is what you guys are doing on your side. What pairs up with G again? So the next one is G. What pairs up with it?
C.
Very good. My next letter is C. What pairs up with it?
G. E.
Very good. My next base is T. What pairs up with it?
A.
Very good. So if you guys ever see that DNA has 1 strand, you have to complete the second strand because we know that DNA has 2 strands. The only one that has 1 strand is RNA. And we'll talk about that a little bit later. So I'm going to give you guys 1 minute. What I want you to do is you're going to make each of these into 2 strands. So each of these should have 2 strands. So that means you're going to figure out what letters pair with it. Are we good with that? I'm going to give you guys actually 2 minutes. So at 3:17, we should be done making 2 strands on all 3 of these. And you guys are doing that on this one right here. Okay? Does anybody need me to repeat the instruction? Raise up your hand. Okay, good. So I'm going to repeat it. So on your piece of paper right here, guys, we're finding the second strand. So for example, you're going to try to figure out what pairs up with G, and you guys told me C. Then you go on to the next space, and you say, what pairs up with G? You guys told me C. Your next space is C. What pairs up with C? You guys told me G. I look at the next space. What pairs up with T? You guys told me A. You're going to finish the second strand for each of these DNAs. That way we actually have DNA. So that's what you guys are doing right now. And I'm actually going to give you 1 more minute. So at 3:18, we should be done with both of these. Does anybody need me to repeat the instructions again, guys? Okay. "30 more seconds, guys, and then we're going to go ahead and get started." "30 more seconds, guys. Finish up." Okay. So I'm going to go ahead and share my screen again, and let me show you guys what you should have gotten. Okay. So what I did here is I finished up the second strand for each of these. So I've identified what is my restriction enzyme. In this case, it is HALE. And now my next one is I need to start cutting in that very specific place. Now here's what's super important. It has to be GGCC. It can't be GACC. It can't be GGCA. It has to be exactly GGCC. So let me show you guys what I mean. I'm going to highlight one of the examples. If I look at it, it matches GGCC on top, and on the bottom, it would be CCGG. So GGCC on top, CCGG on the bottom. And what I'm going to do is I'm going to look at my DNA strand, and everywhere I see GGCC, I'm going to highlight it. So that's what you guys are doing too. Everywhere you see GGCC, you're going to highlight. Now, some kids are like, oh, do I have one here? GGCT? No, you don't, because this does not match. It had to be a C. And because it's not a C, you cannot highlight that. So what I want you guys to do is you're going to look at suspect 1 and suspect 2. And every time you see GGCC in that order, you're going to highlight it. Does anybody need me to repeat the instructions again? Okay, very good. And I'm going to do the same thing on my end. So every time I see GGCC, I'm going to highlight it. So here's another one. Ooh, that's where my mistake is. Yay. Okay. Sorry, guys. I was trying to figure out where my mistake was on this one the entire time. Okay. Okay, there it is. And you might wonder, like, sometimes it doesn't look aligned, and that's just because the way it types out, okay? Don't worry, you have the good amount of base pairs. Okay, so all I'm doing is I'm highlighting my GGCC, and you're going to do that to both of them. And on the bottom, it should be CCGG. Then I'm going to look to see if there's any more. Here's one more. And that is it. Okay. So now I have highlighted the exact area that the restriction enzyme wants to use. Now I have to make my cuts. And in real life, like literally the restriction enzyme cuts the DNA, but on a piece of paper like this, you want to show a cut using either a space like this, or you can add a line. Let me show you guys what I mean by a line. Let me just do this. So you could go to insert and a shape. So you can either add spaces or you can add a line, whichever one you prefer. It is up to you. Okay. Oops, sorry. And what this red line is showing is that I cut the DNA in that specific part. So literally I'm cutting it there. So you guys are acting as my restriction enzymes. So you can either add spaces, or you can draw a line, or you can do both, whichever one you prefer. Any questions about this so far, guys? Any confusion? Now, remember, guys, the way that you get these DNA sequences is usually in real life, they'll send out like a little test tube. You spit into the test tube, or they'll do like a nose swab. Then they take the restriction enzyme. It's usually like a little liquid. They drop it into the pipette. I'm sorry, they drop it into the little test tube. They shake it, and then the restriction enzyme starts cutting up the DNA where it wants it to be cut. Any questions about this before we move on? Okay, cool. So now what we're going to do is the last step is counting our base pair. So Eddie had told me a base pair example is like A and T. Now I'm going to take a little box. You don't have to do this. Actually, I'm going to take a highlighter. You don't have to do this, guys, on your side, but I just want to show you. And I'm going to use green to show you what a base pair is. This is a base pair. G and C. That is one base pair. I'm going to count how many base pairs are in each fragment. Now, I want to show you guys what I mean by fragment. You don't have to do this either unless it helps you. I like doing it because I feel like it helps me, and I hope it helps you. I'm going to highlight what a fragment is. This is a fragment. I'm going to highlight another fragment. This is a fragment. This is a fragment. So can somebody help me out? How many fragments do I have in this first main sample? So how many fragments do I have in this sample right here? Henry, are you there?
Yes, ma'am.
How many fragments do I have in the sample right here?
3? Or...
Yeah, very good. I have 3. 1, 2, 3. So the next thing we're going to do is we need to figure out how many base pairs are in each fragment. So you're going to count how many base pairs are in each fragment. And then I want